Okay, perfect timing. So it's really my great pleasure to introduce Claude Desplan here for today at McGill. Really, and Claude has been a pioneer and inspiration in my own field of Evo Vivo. He's been really somebody who was able to work with the model systems, but then somehow export his knowledge of the model systems out into sort of the natural world and sort of have this kind of dual career of Evo Vivo and Drosophila genetics. Claude basically has, his work has always been inspiring, and he just, about, some, I don't know exactly how long ago, but he pioneered the use of Drosophila for understanding how vision works, motion and color vision. So, and the work has been quite prolific. And along the way, he's actually demonstrated some really general principles in biology, and the ideas of stochasticity in development, and also in terms of evolution, how regulatory systems can change and be stable at the same time. So Claude has really illuminated these kinds of general principles for us, and he's just a dynamo as a personality as well. And so, it's always great to have him around when you're feeling down about something. It's always good to have him around to pick it up and really get you excited about what you're doing. Okay, so, Claude's CV is kind of an exercise of the French academic system. It's a long list. I don't know if I can get it all, but he starts as an undergraduate in the Normale Supérieure in St. Cloud in France. He then becomes an élève professeur, and then agrégation, and then a graduate student in the University of Paris 7, where he worked on vitamin D-induced calcium-binding proteins. So it just goes to show you that academia is a long and winding road until you find your true calling. And then there's a series of assistants and maîtres de conférences until he gets to his postdoctoral fellow at UCSF under Pat O'Farrell. And then he then began his academic career at the Howard Hughes Medical Institute, Rockefeller University as a Howard Hughes medical investigator, assistant and associate professor, and since 1999 he's been the Silver Professor of Biology at the New York University, and also associated with NYU Abu Dhabi. And recently, well, not so recently, but he also got the Sokol Award in the Sciences. And so with that, I'll just give a big round of welcome to Claude Desplan. Thank you.
Okay, so it's a pleasure to be here, and in this auditorium I just realized that you are all subject of Her Majesty. Look very British anyway. So Ehab asked me to talk about most of what we do on basically organismal biology, which means I'm going to talk about the work of one person, and I have to leave behind the work of another 12 or 15 people in the lab who mostly work on fruit flies. But it's actually a very nice story, so a few nice stories, and hopefully you will like it. So, as Ehab says, I have some connections with NYU in Abu Dhabi, which is basically a branch of NYU in the Emirates. And last time I went there in September, I actually went to stop in a Saudi, not a very touristic place, but I went to do snorkeling there, and I just noticed actually that the world of the water is actually amazing. It's not a picture I took to the web. So, it's an amazing colorful world, and the reason why those fish are so colorful is because in fact they have extremely good color vision. The fish retina is made of these 4 types of photoreceptor: blue, UV, green, and red, and they are organized in this beautiful array of photoreceptor, and basically those fish are tetrachromatic: UV, blue, green, and red. And that's why basically the world of fish is okay so rich. But then what happened when the fish came out of the water and they ended on land? The world was run by dinosaurs, actually them outside, and when you are in a world run by dinosaurs, what you tend to do is to live at night, stay small, and when you live at night you lose your color vision because cones cannot be stimulated by low light, and as a consequence most of the mammals actually colorblind. Your mice and rats and cats and rabbits are all colorblind, or at best dichromat. And what happens if you see the bull being fought by the bullfighter, he sees a red cape, but the bull doesn't care about the red cape, he just sees something in front of him. So this was the mammal, but then something happened about 5,000 years ago. I mean, no, not exactly. So Eve wants to give the apple, and of course Adam needs to see the red, and therefore they have a red color photoreceptor. So, I mean, everything was very planned. It's like a pre-adaptation. It's called pre-adaptation. So pre-adaptation is like 20 million years before. What happened actually from this dichromat, nature invented actually another pigmentation, the red pigment, and basically allowing the spectrum to expand into the red. And as human now, we are all trichromats. Okay? And we are trichromats. In fact, we have 3 types of cones. It's actually a picture taken from a live human being with an adaptive optic. And what you have here, 3 types of cone: blue, green, and red. And what you can see with now you are a trichromat. Now, what the specific feature of this retina is kind of strange because it's compared with what you saw with the fish retina. The fish retina had 4 types of cones. This one was only 3. But the fish retina is also very beautifully organized. Blue, UV, green, red, blue, UV, green, red. This one is a mess. Okay? You have proportion about 7% of blue, some green, some red, and you cannot predict who is going to be what. What this retina is actually stochastic, which means you cannot predict what's going to happen, what photoreceptor is going to be at this location. And therefore, it's going to lead to some trouble in order to be able to interpret the image coming from that, because the brain will receive an input from a photoreceptor. You will not know exactly where, which color it is. Anyway, so this is what happened in human. Some of the male, about 7% of the male population is colorblind. It means they lost one of the photoreceptor, and they go to the farmer's market instead of looking at the fruit like you see here. They see it that way. You lose quite a lot of information. Still, we can manage to survive. The fruit is still quite good. So if Adam had been like that, maybe we would not be where we are. Okay, so now I want to go back to this notion of stochasticity that Ehab mentioned. And actually, I want just to give you a famous quote by a famous man, which is basically that he does not play dice. It means that nothing is left to chance. And if you look indeed at triplet or quadruplet, who basically has the same genome, they're extremely similar to one another. Okay? And so indeed, it seems that he doesn't play dice. I'm very religious. So he doesn't play dice. In fact, everything is in our genes, and nothing is left to chance. Now, this man, who most of the time did get it too much wrong because it's Einstein, he didn't do what you should do, or is actually read all papers. There is a lot of things you can read in all papers. In this case, he didn't read an old paper from 2,000 years before in the Talmud, which said everything is always foreseen, but there is still some freedom of choice which is left to you. So what this guy meant actually is that what was going to happen a few years before, after that, is that sometimes nature indeed just leaves some room for chance. Exactly, it's a bacterium, Bacillus utilis, and this is an example where Bacillus utilis, of course, all the bacteria are the same because they're all sisters from one another, and they should all be the same, but you can see here some of the bacteria are red and some of the bacteria are green. The bacteria which are green, they are green because they express a transcription factor called COMK, which makes those bacteria competent to receiving DNA. Which means those green bacteria will be prone for infection by virus or whatsoever, and most likely they will die, while the red will survive. But in case you treat this bacterial solution with antibiotics, maybe the green one will be able to capture some antibiotic-resistant plasmid and will be able to survive. So it's some way of like hedging your bets, where you just set aside some bacteria which most likely will die, except there is some circumstance where they can be able to evolve. So people thought for a long time this was reserved to the bacteria, but in fact we know there is a lot of this stochastic decision also made in vertebrate, in mammals. And maybe the best example, the one that makes a lot of sense, is what happened in the olfactory system of the mouse of human. And as you might know, among the 25,000 genes in our genome, about 1,400 are dedicated to being olfactory receptor, which means the biggest gene family in our genome is olfactory receptor because each one of them needs to detect one specific odor. And in sensory perception, there is one general rule that must apply at any cost, is that once you decide to be a receptor for example, alcohol, you can only detect alcohol, you cannot detect aldehyde and fatty acid whatsoever. So you can only express on your surface one of the 1,400 olfactory receptors. And if you have to do that for 1,400 genes, it would be an immense work to be able to find a precise way of putting this receptor here, this one here, this one here. So nature instead evolved a system whereby it says, okay, you choose which receptor, which one do you want? A, okay, you express A, but you exclude all of the others. You choose D, okay, you choose D, but you exclude all of the others. And therefore, now what you have, a stochastic distribution of photoreceptor, each one of those 1400 is expressed in a stochastic manner in a nasal epithelium. Now, you have also some other example of stochastic choice. Actually, the butterfly's wing, and there are scales of the butterfly wings, and you can see each one of these scales make a stochastic choice to be blue or to be black. And actually, this stochastic choice in this case can be very dense here. It can be much more sparse here. And again, here you can see some more example where you can see again, you can make some stochastic choice. You cannot predict what happened, but you can still control somehow the ratio of those different stochastic choices. And of course, your footprint or fingerprint, sorry, can also be completely statistic. We don't understand how this is being made. But I want to go back to this notion that this happened actually in the context of the human retina. And the question I want to ask is, okay, now we have a stochastic choice here, and we can ask two questions about that. So first, why is there a stochastic choice here? And maybe just a fluke of the system, and also how this system occurred. So, in fact, we kind of know we don't know the why, we know the how. In the case of the human color vision, as I was telling you, what happened is that in the mammals, we were only dichromat as mammals, blue and red photoreceptor cells. And I'm going to ignore the blue and only keep the red. And what happened is actually in the lineage leading to human, there was a duplication of the red gene, and now we have two red genes. And the point actually, those two red genes, one of them got tuned to becoming green. So now you have two genes next to one another, but in the process of doing that, nature forgot to duplicate the control element that controls these genes, and there is only one of them for two genes. And this element can only talk to one of the two genes, which means either choose the red gene here, or you get to choose the green gene here. And this is how you get a stochastic choice. Because in fact, you can either get green or to be red, and therefore you make a choice. Now, there's a problem in this model that in fact, one allele could choose to be red, the other one could be green, and then you would disobey the rule that I established at the beginning. Not so, because in fact, these are like X chromosome, so boys don't have any trouble because they have only one X, and girls have X inactivation, and as a consequence, in every cell, only one of the X is being conserved. So now we know how this happens, but now what about the why? So why is more difficult to address, and maybe why the stochastic choice occurs simply because it was the best way in nature to expand the spectrum of color discrimination by adding one more photoreceptor, and the best way we could find is to do it in a stochastic manner. But maybe we can actually look outside of human and maybe try to get some reason for that. So we were actually interested in this process because we were working on the fruit flies, and we work on the fruit fly eyes, which obviously is a much better system to study than the human eyes. And the fruit fly eyes, as you might know, you will see has a lot of similarity with the human eye in terms of this distribution of photoreceptor cells. So the fruit fly eye is a compound eye, which means it's made about 800 unit eyes. Each one is its own lens. Each lens is about 1 pixel. And therefore, the fly eye is about 800 pixel resolution, very low resolution. But when it moves, actually, it can actually improve this resolution. Under each one of the pixels, there is about 8 photoreceptor cells. There are 6 rods, black and white, motion detector, height sensitivity. We're going to ignore them and focus on the remaining 2 photoreceptors, R7 and R8. You only see one of them because they are one on top of each other. And that actually is the cone of the flies because they express one of 2 rhodopsins. The top one expresses UV opsin, and the bottom one expresses blue opsin. It's called R7 cells, called R8 cells. They express UV and blue, and therefore, when the light comes through, it goes through the UV, then to the blue, and therefore, the animal can compare between UV and blue and achieve color discrimination between those 2 types of color. In fact, the story is a bit more complicated than that because there are, in fact, 2 types of ommatidia of unit eyes. There are those that make the UV blue comparison, and those that make the UV green comparison. So it means, in this case, they compare between the longer UV and green, which means they discriminate among longer wavelengths of light. And the funny thing is that, as in the case of the human retina, those 2 types of photoreceptors are distributed in a stochastic manner. What I mean by that, actually, if you look at the green and the blue opsin, what happens, they are distributed like that in a random manner. And maybe if I'm going to show you both what happens in green and blue, and the 2 types of UV, what you have in the next slide. So what you have here, a proportion of the R8 photoreceptors that express the green opsin, the remaining express the blue opsin, 30 to 70% ratio of the two. Same ratio exists in the cell on top, the R7 cell, the two Uveopsin, rhodopsin 3 and rhodopsin 4, 30-70. So you have two stochastic distribution, but in fact there is only one of them because you always find the short UV with blue and the long UV with green. So now what we have a problem is that first, again, same question as before, how does this happen? And second question, why does this happen? So, in fact, how this happened came quite a while ago when we actually discovered something. We discovered, so basically the fly is a double dichromat in the sense it compares UV and blue and UV and green. So it's kind of a trichromat. Okay, so we wanted to investigate what was happening, and so we got some understanding what was happening when a number of years ago we cloned a gene, a transcription factor gene, which encodes a gene called Spineless. It's a BAS-BHLH transcription factor. It's a gene that binds to DNA. And this gene is expressed extremely early during development in the photoreceptor cells. And you can see here, it's so early, at least the 8 photoreceptor cells are not even photoreceptors, they're just cells which are being recruited for becoming photoreceptors. They know who they are, but they have no sign of differentiation whatsoever. They will only differentiate in about 2 or 3 days. And one of the cells here, always the cell expresses the transcription factor Spineless, and this cell is actually the R7 cell. One of the cells makes a stochastic choice. And in fact, what happened, this transcription factor Spineless is expressed in a subset of the R7, is not expressed in the other subset. Which means what you have 70% of the R7 cell very early in development express Spineless, the remaining 30 don't express Spineless. And this is exactly the proportion of the photoreceptor, those same R7 cells, that will later on express rhodopsin 4. So if you look in the adult, the gene is maintained in the adult, and what you have here, 70% of the cell R7 cell express rhodopsin 4, the remaining express rhodopsin 3, and maybe this is exactly the same as the Spineless gene. So now what happens if you remove the Spineless gene? We only remove it from the eye, not in the rest of the body. If you do that, everybody gets transformed into the non-expressing Spineless, the rhodopsin 3 cells. And if you force expression of Spineless in all of the 100% of the R7 cell, now everybody expresses rhodopsin 4, which means the Spineless gene is both necessary and sufficient for controlling the stochastic expression of the rhodopsin 3 and rhodopsin 4 in the R7 cell. Now again, to say big deal. We started from a stochastic distribution of rhodopsin into R7 cell, and now we understand it's due to the stochastic distribution of the Spineless gene in the R7 cell. But we moved at one step. But who is controlling the stochastic expression of the R7 cells? And I don't want to give you the answer because we know quite a lot about it, but it will take the whole seminar. What I can just tell you is that nobody tells Spineless to be expressed in a stochastic manner. Spineless decides on its own to be on or to be off. And a former postdoc of mine, Bob Johnston, he studies that in his own lab at Johns Hopkins, and we know exactly that the gene just decides to be on by folding its chromatin in a specific way, to be on or to be off, and then it remains stable that way. Okay, so now that we have a gene, and this gene is expressed in a stochastic manner, is one of the two R7 cells, and this gene is a transcription factor. In fact, it binds directly to the promoter of rhodopsin 4, which means if you express Spineless, you express rhodopsin 4. If you don't express Spineless, you don't express rhodopsin 4. Spineless also turns on a repressor called DVE. DVE is a gene, a transcription factor, which also is a transcription factor, which also binds to DNA, and now it binds to the promoter of rhodopsin 3 and repress expression. And therefore, if you have Spineless, you don't have rhodopsin 3, you only have 4. If you don't have Spineless, you only have rhodopsin 3, you don't have 4. Okay? Now, what about the R8 cell below? I told you there is coordination between R7 and R8. In fact, what R7 does, it prevents a signal, an activin signal, that goes from R7 to induce rhodopsin 5 expression. And in the absence of the signal, you get rhodopsin 6 by default, and then those two cells, those two genes actually repress each other. And at the end, what you have, the nice situation where you have 4 and 6 in R7 and R8, 3 and 5 in R7 and R8. And basically, you have your stochastic choice in both R7 and R8. Okay, so this is all very nice, but the question that doesn't tell us why does nature choose to express those genes in a stochastic manner. So maybe some things that can happen, maybe it's because they don't know how to make it in a deterministic way. They don't know how to make stripes or row whatsoever. So we were thinking about that, and then one day we just received a phone call by a person, a lay person in Chicago, and he told us about those flies called Dolichopodidae, which are fairly related to the Drosophila, but still fairly far in terms of evolution, about 160 million years ago. So Keith Short, this guy, is actually an amateur photographer. He's actually a mechanical engineer. He built the gearbox for the 787. So, and the problem, he loves to take pictures of insects in his garden. His wife believes it's crazy. And he takes those beautiful pictures of these Dolichopodidae flies. So he sent me an email one day and said, Dr. Desplan, I know you're interested in stochastic choice in insect eyes. What do you think of this picture? So this is the eye of a Dolichopodidae, and if anything but stochastic. Beautiful rows of this yellow and red ommatidia. And again, I'm not talking about rhodopsin here, just the coloration of the cornea. Still, what it tells you is that these ommatidia here, which are all these rows of red, have something specific which is different from the next row. And for those who know how the fly eye develop, you might know they develop from an epithelium. There is a flat epithelium, and there is a wave of differentiation that goes from the posterior, move to the anterior, and in the wake of this wave of differentiation, you get basically recruit all the photoreceptor cells. Okay, it goes from right to left, you recruit them. In fact, on the next picture, you're going to see beautifully how this happens here. So actually the wave was migrating from right to left, and what happened at this stage, you start making a lot of mistakes. Maybe when the furrow was migrating at this stage, it was desiccated or it was heat shock whatsoever, you start making a lot of mistakes, and a single mistake has no consequence. When you make 2 or 3 mistakes, now you switch the rows, and now you go to the off rows to be able to. And you can see here, then the thing went back to normal, and it gets propagated normally to get back. Of course, there is a problem here when those 2 rows propagate, and this tells you something about the mechanism of propagation of the formation of those 2 rows of cells. So we say, okay, this means that in fact, the insect know how to make beautiful rows and stripes, but does it correspond to actually rhodopsin expression, which is underneath? So we sequence the genome of these flies, we clone the gene for rhodopsin, and actually there's a picture for the blue rhodopsins, and we made antibiotic against this blue rhodopsin, and you can see that in the RH cells, you have blue, green, blue, green, blue. I don't have the green here. So you have basically an alternance of blue and green, which means what you see on the surface is reflected inside of the eye by the expression now of rows of rhodopsin 3, of green and blue rhodopsin. So again, here is a large spectrum. In red, you have the blue rhodopsin, sorry for the code. And actually, I didn't stand it because it's a fairly new result. In green, what you have is the other rhodopsin. You have beautiful arrays of blue, green, blue, green, blue, green, blue, green. So indeed, flies know how to make stripes. And the funny thing actually now is this is actually the ventral part of the retina. If you look at the dorsal part of the retina, then it becomes stochastic. It means those rows, they continue, but they get interrupted in a stochastic manner. And so basically what you have is basically something that looks much more like a fly retina here as compared to this the retina here, which is very organized. So we don't exactly understand why those flies, and only those flies, chose to be deterministic as compared to most of the other flies which are completely stochastic. One explanation may come from the following. So again, I'm back here at a fruit fly, the Drosophila eyes, and it looks very homogeneous by scanning here. But in fact, you remember this eye is made of this stochastic choice of 2 types of ommatidia, the UV blue or the UV green. But it also has here, at the dorsal edge, one single row of ommatidia here, facing the sky, okay? This row is unique row, and this unique row is very special because in fact, instead of expressing rhodopsin 3 and rhodopsin 5, UV blue or UV green, it expresses UV, UV. Both R7 express UV and UV. If you express UV and UV in R7 and R8, it cannot serve for color discrimination because there is no purpose. In fact, what this is something which is, so again, you can see now with the molecular marker, this is a stochastic distribution of Spineless in the main part of the retina, and at the edge, both R7 and R8 express another transcription factor called homothorax, HTH, and homothorax is a gene that specifies this very special type of ommatidia, and what the purpose of this ommatidia is in fact shown here, exactly what I showed you before, the 6 outer and the tiny R7 cell. They don't care about detecting many photons, they just want to be able to discriminate the wavelength. They're going to be UV or rhodopsin 4. But in the dorsal margin, in this part of the eye that faces the sky, the photoreceptors are much larger, they receive much, much more light. And these are the ones that express rhodopsin 3 in both R7 and R8. And what these are actually, they serve for the detection of the vector or light polarization. So it's a view of the world from an insect's point of view, and basically they don't see very much. They see a bunch of sky, a little bit of sky, blue sky, and they need to know where to go. Because, for example, if you have monarch butterflies, if you want to migrate, you want to know exactly where the sun is because you want to know where Mexico is, and therefore you know where south is, and so if you don't only see that, you're not going to be able to go very far. But in fact, the sky, as the sunlight is reflected by the sky, and the sky light is actually polarized, which means in fact, even if the insect, the butterfly, the fly doesn't see the sun, he knows what the sun is because the light is polarized into one specific direction. You know what polarized light is? Light is unpolarized, it vibrates in all directions in space. When it goes through reflection on a non-metallic surface or on the sky, it only vibrates in one direction. And when you use your Ray-Bans, you actually only choose to have one orientation of the light that can go through. And what you have here in our system, we have photoreceptors which have polarization. They are actually membranes which are like very regular with wall-to-wall rhodopsin. And the R7 are made like that, the R8 are made like that. Basically what you have two crossed over polarizing filters. So what this dorsal margin area in a fruit fly is actually a polarizing detector. They can measure the vector or light polarization. So the reason why that rhodopsin 3, the UV rhodopsin, both R7 and R8, because they don't want to confuse color discrimination and polarized light discrimination, which means they need to be monochromatic in order to be able to see that what they see between R7 and R8 is actually the difference in polarization. Now, the funny thing is that in the Dolichopodidae flies, the flies I just described to you, you remember you have these stripes, a red stripe and a green stripe, red stripe and a green stripe, and in the red stripes, the photoreceptors are almost like in fruit flies. That means the R7 cell, which is here, it sends this microvilla like that, and the other one, let's forget about them. And basically, the one below will have R8, will have orthogonal orientation. It's going to be something very good at detection of the vector light polarization. The funny thing is the other row around, the next row over, instead of sending the microvilla like that, it does that. It just makes it, which now the R7 and R8 will have the orientation of the microvilla parallel to one another, which means in fact this is going to be a polarizing filter, not something that allows you to figure out the vector or light polarization, just something that cuts off polarized light. Those flies live on very shiny leaves, which is very polarized. Maybe this helps increasing the contrast by doing this kind of thing. Okay, so what we have here, we have basically the idea of that nature as using a lot of features to be able to adapt the world that he sees, his vision to the world that he sees. But we are not very satisfied because now we know that the fruit flies can have a stochastic distribution and that Dolichopodidae have a non-stochastic distribution, but maybe it's just a fluke that the fruit flies that made this stochastic distribution. How far away can we see this stochastic distribution if you look in a phylogenetic tree? So we decided to look in butterflies because, as you might know, butterflies actually are relying a lot on color vision. Often you have several hundred of species of related butterflies in the same area, and they need to be able to find their mates. And often the only thing they can distinguish from one another is actually the color of their wings. And those butterflies also rely a lot on flowers to be able to feed, and therefore they have extremely good color discrimination, color vision. So in collaboration with Kentaro Yarikawa in Japan, we decided to look at the visual system of the butterflies and to try to analyze what was going on there. Okay? So we had some indication that the butterflies might also have stochastic distribution, because when you shine some light onto a butterfly eyes, it's a fairly complicated system. You get like a mirror in the back of the eye that reflects the light. You can see actually that the eye is much larger than many more ommatidia. It does reflect light in different ways, and you can see clearly there is stochastic distribution of photoreceptor here, but we don't know what this means. At least there is a mirror there that reflects light in a different way. So now I'm going to give you some background on the butterflies. So first in flies, you remember you have two types of ommatidia or unit eyes. The one express UV and green, the one express UV and blue, and basically it's double dichromatic. Butterflies are a lot more complicated. So in fact, there are three types of unit eyes, and those three types except five types of rhodopsin. So I'm going to go slowly. Those two photoreceptors here always express the same green opsins in all three types. This one expresses either blue or UV. This one expresses blue or UV. Okay? And the four bottom ones express either a dark green opsin or a red opsin or a mixture of a dark green and red, which means in fact, as you will see, the dark green is expressed at the lower level. So you have three types of ommatidia that contain five types of rhodopsin, and the question we can ask is, how does this happen? How do they build this retina, which is much more sophisticated than the fruit fly eyes? And how can we, using the knowledge, the deep knowledge we have from the fruit flies, how can we figure out what happened in the context of the papilloid? So I want just to focus your attention simply on two photoreceptors, this one and this one here. So this is actually going to express blue, blue, blue UV or UV, UV. You have a picture here. You can see here, you can express, so sorry for the color code, you have blue UV, 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 blue UV or UV, UV, or blue, blue, okay? So you have three subtypes of here, it can be blue, blue, UV, UV, or UV, blue, or blue, UV, okay? So basically now, I'm just going to focus my attention on these two photoreceptors, blue, blue, blue UV, or UV, UV, okay? And to some extent, it's quite reminiscent to what we saw in the fly retina, because here what you have a stochastic shot of expressing UV, rhodopsin 3 or rhodopsin 4. Here you have, so it's, and remember, this one corresponds to the expression of spineless, so it was spineless on, spineless off. Maybe you can imagine you have the same situation here. Maybe this is on, off, off, off. But how does this mean? How does it mean? So one solution would be that simply, instead of having one R7 cell, as in fruit flies, what you have in papilio, you have two R7 cells, and each one is allowed to make a stochastic choice. And this will fit perfectly well the system that you have here. Remember, butterflies and flies are 250 or 300 million away from fruit flies. They are very, very far away, but maybe they conserve the same pathway and are able to basically expand their color vision spectrum by maybe increasing the number of R7 cells that make stochastic choice. So let me give you some background to be able to see whether this is true or not. So exactly the fruit flies markers. Exactly markers, the spike gene marks the two R7 and R8. Prospero marks only R7 cells. Spineless marks only the R8 cells. And of course, Spineless marks only a subset of the R7 cell. So we have four genes that allows you to basically figure out what happened in the context of this color vision system of the fruit fly. So we sequence the genome of two butterfly species, Papilio and Vanessa, and we made antibodies against Spal, Prospero, Spineless, and Spineless in this species of butterflies, and we looked at what happened in the context of the butterfly eyes. So in the fly eye, remember Spal expressed in R7, R8, and what happened in the Papilio? In fact, in Papilio you have three photoreceptors instead of two that express the Spal gene, which means you have three color photoreceptors instead of two. You can have two R7, one R8, or two R8 and one R7. So now what we can do, so something like that, and actually this cell here at the bottom is actually another, so the three cells, two here and one here. Now we look at senseless, the one that marks the R8 cells, and R8 cell in butterflies appear to be this cell, the one in the middle. But now the question remains that most likely what seemed to happen, our hypothesis was true, so maybe we have two R7 cells. And remember, R7 cell is the cell that makes the stochastic choice in the fruit flies. So we have two R7 cells. So we mark for the Prospero gene in marker 7, and indeed you have two R7 cells. So what you have, the reason why you have expansion, actually you have two R7 cells and one R8, and maybe indeed what the model was true, that indeed those two R7 cells can each one make an independent choice. Now the question is, you have now the possibility of making this choice, but is the spineless system conserved? So in flies, what you have spineless is on, off. You could imagine the spineless on, off,